Okay, if you guys can't see my screen, let me know. But you should be looking at PowerPoint. Okay, so
It's a little small.
what about now? Should be like full screen.
Yeah, it's full.
Okay. Starting Monday, I'm gonna be going back to campus and like lecturing in a classroom on the whiteboard. You can still, if you're doing okay, you could still stay home and do it that way. It's just, I'm just leaving it up to you. We're supposed to stay off campus till February 1st. So I should have probably went back today, but I got my other COVID shots. So I thought I was going to feel bad this morning. So I just did it this way, but I'm going to go back to I'm going to go back into the classroom. I'm still going to use like PowerPoints and stuff. So, I mean, it really doesn't change anything for you. Just if you decide you want to come on campus for anything, I'll be there. That's the only thing that's changing. So, yeah, so today we're finally on something that has to do with biology. And biology, it's just something living, and something living has to be able to replicate. So, we're on cells, and cells are the smallest thing that is living. And there are two types of cells. So there's one way that we can like delineate cells, and we can call them prokaryotic and eukaryotic cells. I know those words kind of suck, but look at this word up here where it says prokaryotic. So this is a prokaryote. And the only thing that you need to remember being a prokaryote is bacteria. There's bacteria, and then there's these things called archaea, but I mean, it's pretty much bacteria. So bacteria are a whole different type of cell from all the other cells that exist. And so this is just this is one of the types of bacteria. So you got on one side bacteria, and you have on the other side these cells called eukaryotes. So instead of being like PRO, it just says EU. And then the rest of the words are the same, KARYOTE. So there's prokaryotes or prokaryotic cells and eukaryotes. Eukaryotes are everything that's not bacteria. So plants, animals, fungus, whatever. That's all sponge. They're all eukaryotic cells, right? So only bacteria are prokaryotic cells, and they differ. The main difference with them is in how their DNA is. So you see here in the middle, their DNA is all just like loosely hanging out in something called a nucleoid. So I'm like circling it right. So they don't have a nucleus. So if you look at like, this is like an animal cell. This is like a eukaryotic cell, right? And so it's been kind of like cut in the middle. So there's a nucleus. It's kind of like a ball. And it's got like all your DNA's inside, and it's got like this membrane around it, right? That's theirs. This doesn't have any membrane around their DNA. Like their chromosomes, they're all just kind of loosely hanging out. So we call that a nucleoid. And so that's the difference between them. That's it. That's the big difference between prokaryotes and eukaryotes. We're not going to spend any more time with prokaryotes. That was it. Just wanted to show you that one slide. Yeah, so what we're going to spend our time on now is eukaryotes. So now if we take all the eukaryotes, we could divide those into two types of cells: animal cells, plant cells. So that's it. We're going to talk about animal cells first, and then plant cells are similar. They just have some they have a few differences that we're going to discuss. But once you know what is in plant cells, animal cells, you're going to know it's in plant cells. So, I know I'm eating on camera. Let me let this person in. So here's an animal cell. So where do we start? Let's start with the middle here. There's the nucleus. It's usually the thing that you see in a cell when you're looking at a slide of a cell. When we're looking at them under a microscope, we see the nucleus, right? So the nucleus has all of your genetic information, all your chromosomes. So right here, they're calling it chromatin. So if you look at the nucleus, there's some different parts of this nucleus. Here's chromatin, where my pointer is. That's all the chromosomes. You see in the middle here, it's kind of like darker. So they're calling that a nucleolus. The nucleolus is going to make something called ribosomes. So we're going to talk about that all later. So this chapter, by the way, there's a lot of words here, and so that's going to be kind of difficult, but it's easy in the sense that it's just like memorization, right? If you like to make flashcards, this is a chapter for you, right? Because you just write on the front nucleolus, and on the back you write makes ribosomes. That's it. And when I ask you on a test, that's pretty much, how am I supposed to make a question on this, right? All I can do is I can say the nucleolus makes blank, or I can say what makes ribosomes, right? It's the only way I can answer it, or that I can ask you. So in that way, yes, it's a bunch of words, but it's just memorization. You don't have to put like the information together. I can't ask you any other kind of question about it. It's just direct. What do each of these things do? And then you should know a couple words, and that's it. So it's just memorization. So yeah, the nucleolus makes ribosomes. We'll talk about ribosomes in a second. And then here's all the chromatin. That's your DNA, or we could say your chromosomes. And then there's an envelope. They're calling it an envelope, but it's the membrane that surrounds it. So it envelops the DNA. So the nuclear envelope is the membrane that goes around it. That's it. So that's the nucleus. All right, so we have the nucleus, and then out here, this white thing, that's the membrane around the cell. We have a whole chapter on that, so I'm not gonna spend time talking about it. We call it a phospholipid bilayer, or another word that they'll call it here is plasma membrane. But that's it, right? So you have this membrane. Inside, in the middle of the core of the cell, you have a nucleus, and then you have everything in between, all this other stuff. So all this other stuff, we call them little organs. So they're organelles. So organelles are just, they mean little organs, right? And so we're just going to learn about all these little organelles and what they do. They each have a function. And so that's what we're essentially doing. We're going to go through them and just talk about their function. There's lots of websites that you can go to where you can click on them and know what they do. By the way, I'm never going to ask you, I know in high school and in some college classes, they'll have a picture and you need to if I'm pointing on this thing, you need to know it's a mitochondria. I'm not going to do that. I don't really care that you know what it looks like or don't know what it looks like. So I'm never going to ask you to pick them out by sight. So here's the nucleus, here's the cell membrane, and all these organelles, all these things in here, they're floating. They're all floating in a fluid. So we call that fluid cytosol. So I'm going to spell it out because I don't have it written here. It's Cytosol. Whenever you see the word cyto, CYTO, in a word, it means cell. Right? So that liquid's called cytosol. Another word that we can use to talk about this area outside the nucleus is we could say cytoplasm. So again, cyto, C-Y-T-O, and then P-L-A-S-M. So we call this area cytosol or cytoplasm. So we can use it to mean outside of the nucleus. For example, we have DNA inside the nucleus, but we make proteins and lipids out in the cytoplasm. So when I say out in the cytoplasm, I'm talking about this area out here, outside of the nucleus. So all these organelles are in the cytosol or cytoplasm. And so let's go over each one of these organelles. These organelles that I have out here. So the first one's going to be a ribosome. Oh, that's the nucleus actually. So let me, there we go. And I've already written on here what they do. A lot of them I've written in. So ribosomes make proteins. That's it. That's really what you need to know. Here's cytosol, and I'm telling you that the cytosol is like the liquid part. Just what I got, I just got done saying that. Cytosol is the liquid stuff that all of the organelles are floating in, right? So in that cytosol, one of the organelles is called a ribosome, and ribosomes make proteins. That's all you need to know about ribosomes. So what are the only two questions I can ask you on this? What do ribosomes do? Or I could flip it and say what organelle makes proteins. That's it. So if you associate the word ribosome and protein, you're good. And you should know that because this stuff, again, most of this stuff, I keep saying this to you guys, but it's true. Most of this stuff is going to come back in another class. So the more you know now, the better. You've got to learn it anyway, right? You've got to learn it for a test. But just while you're learning it, keep it in your head that this needs to be like permanent memory. You don't want to just learn it just for the test and then forget it. That's not doing you any, it does you good because you can pass the test, but it's not going to help you later. And you will come across all these things. For example, if you take microbiology, you're going to talk about ribosomes a lot. If you take pharmacology and you're learning how antibiotics work, again, you're going to be talking about ribosomes. So some of the ribosomes are floating. So you see how I have here circled free and bound? Some of the ribosomes they look like little dots, right? And they're just floating around. So like you see, well, there's no good photos, but there's like little specks of grains in here. They're ribosomes, and they're making proteins. But some of the ribosomes, if you look at this blue thing, you notice that this blue thing, some of this blue stuff has red dots attached to it, and some of this blue stuff doesn't have any red dots, right? So all this blue stuff in here, we call it ER. You don't have to learn endoplasmic reticulum. You could just say ER because that's what everybody says, right? But there's two types of ER, rough and smooth, right? So some of this ER is smooth. It doesn't have the ribosomes. That's what the little red dots are. Some of it does not have ribosomes. Some of it does, right? And so the ER that has ribosomes stuck to it makes proteins. because ribosomes make proteins. Some of the ribosomes are floating around by themselves. They make proteins. Some of the ribosomes are stuck onto this blue thing called the ER. It makes proteins. That's it. Ribosomes make proteins. That's what I'm saying here is that like some of them are attached to the ER. Some of them are floating around free. In either case, doesn't matter. They're making proteins. Let's talk about the ER now. Endoplasmic reticulum. It's the thing, it's right outside of the nucleus. So we have the nucleus here, and usually right next to it, like really close to it, is the ER. I already told you some of it is rough. The rough ER makes proteins. Smooth ER, which does not have any ribosomes stuck on it, makes lipids. So there you go. So these are like flashcards, right? Smooth ER, flip the other side of the flashcard, makes lipids. I bet if you look on Quizlet, you'll find these already done. Or again, if you go to a website like Cells Alive, you could probably do it even faster. You know, you just click on things and it tells you what it does, and you just start learning it that way. So smooth ER makes lipids, rough ER makes proteins. You guys have any questions so far? Am I going too fast?
Will you ever be that teacher that posts a picture and says just fill in a blank?
Never. I hate pictures. I don't know if you guys like them, but I mean, I don't see the use in them. I'll never make you color anything either. I hate coloring with colored pencils and stuff. If I'm going to give you points, I'd rather just give them to you. Because that's all the teacher's doing anyway, right? That's why you're doing it, because they want to just give you points. Okay, fair enough. Just give you the points. They do that to our kids. They make them do those stupid posters. I don't know if you guys have kids and you have to do those stupid posters. Just give them the points. Don't make us suffer. You know the parents are doing the stupid posters. Wasting our time. Kids aren't learning anything from that. And everybody knows it. All right. Sorry. All right. More than a few words here. The Golgi. Just remember the word Golgi, and that's fine. Because they call it different things. Golgi apparatus, Golgi complex, Golgi bodies. Just remember the word Golgi. So it's like if you work at, it's like when you work at Amazon, right? You don't stick everything in the same box, right? If you have something like super long, that takes a different type of box than if you have like a coffee mug, right? So you can't, that's what Golgi does. It takes the proteins, because proteins are going to be different things, right? Some proteins are going to be hair, muscle, but some proteins are going to be just like antibodies or something like that. They all go in different packages. So the Golgi takes different proteins and lipids and packages them different ways, depending on what they're going to eventually do. So it modifies and packages proteins and lipids. And it can make carbohydrates. Right, so we have our three macromolecules here. We have proteins, carbs, lipids. The cell can make all of these. So again, you don't have to know what it looks like. You know, although I'm going to tell you one thing that has nothing to do with the test. Trying to find a nice picture. You know, when we discovered electron microscopy, and it wasn't that long ago, like my mother would have never heard of it. She would have never seen a picture of it, never really had access to it as a kid. but it really made a difference in what we were like. When my mother went to school, they didn't talk. They didn't have any idea about all these things inside of a cell, right? Because we didn't like the technology wasn't being used. We didn't have this technology. It kind of was around, but it really wasn't used. When this technology came out, it really allowed us to see so many things that we had no idea, right? Because no microscope, no regular microscope, no matter how good it is going to be able to get these pictures off of a nucleus. You're not going to be able to see the little holes in the membrane. You just aren't. All you're going to be able to see is a purple blob. And it doesn't matter how close you can look, with the best microscope, you can't see it. But these electron microscopes, I don't know how they do it really, but they're able to get in there. So it makes a huge difference in what we've been able to see. And I know these are never really impressive, are they? It's just like black and white shots of nothing, right? But it allowed us to understand that there's these things called ribosomes that make proteins, right? And then we start to look at the ribosomes and say, well, can we manipulate them at all? Can we stop a bacteria can we stop their ribosomes from working? Because if we can, that's an antibiotic, right? But we weren't able to figure that out until we had this microscope come. So when you look at this Golgi apparatus, it does not look like, it doesn't look like anything really. I wonder why they put them in the book, because they don't look like anything, do they? But nevertheless, it was a really profound discovery in science. So Golgi repackages proteins and lipids, and it can make carbohydrates. Okay, lysosomes. I have 3 slides on this. We don't really need them all, but here I'm going to give you the key word right now. Lysosomes contain digestive enzymes. Right here. Lysosomes contain digestive enzymes. Remember, enzymes, any reaction that you have, catabolic or anabolic, needs an enzyme. So whenever you build something or whenever you break something down, you need an enzyme. In this case, digestive enzymes, are they for anabolic reactions or catabolic reactions? And you can just think to yourself if you want. They're breaking down something. So catabolic, right? Digesting things, those are catabolic reactions, breaking things down. We did that lecture. Okay. So that's what you need to know right here. Contain digestive enzymes. That's the only takeaway that you need to know for a test question. And you can just put it down just like that, and I'm fine with it. What are lysosomes? And your answer, contain digestive enzymes. Or you just put digestive enzymes. Good. That's it. That's the answer. So here's a piece of food outside of the cell, and the cell is going to take it in. And notice this word. This word is in the next lecture, but it's called phagocytosis. But already you should know something about this word. Look at the word cyto, C-Y-T-O. So already, if you're just looking at the word and you don't know anything about it, you see the word C-Y-T-O, and you're going to think cell. Okay, that's something to do with cell. Then we have this word phago. So this word is in the word esophagus. So P-H-A-G-O means to eat. So already the word, we know what it is. We know what this word phagocytosis means without even hearing it before. Phago means eat, and cyto means cell. So it's a cell eating. That's the process of the cell eating something. We call it phagocytosis. Or if I want to tell you about a cell that eats things, I'll call it a phagocyte. Anyway, so take in the food. Here's a vacuole. A vacuole is just like a bowl that you put the food in. It just carries different things. And then here's the lysosomes that meet it. And the lysosomes are, they're going to digest the food, and that's it. At the end of this, once the lysosomes are done with the food, like let's say it's glucose. Once the lysosomes are done with it, it's not going to be anything anymore, just atoms. Anyway, take away from this, lysosomes contain digestive enzymes. That's it. Mitochondria. So it's two words. Mitochondrion, like it's written up here, is singular. Mitochondria is plural. I don't care if you know that or not. It doesn't really matter. Just you'll see it written in both ways. So just know it's the same thing. It's not some different thing. So the mitochondria is where ATP is made. So we talked about last class how ADP is put together with a P and you make ATP from it. This is where it happens. And so we have a whole lecture that just talks about that, how ATP is made in the mitochondria. And it's an interesting organelle. It's an interesting organelle because, look, it's got like a membrane here, and it's got another membrane in the middle. So it's got like these two membranes in it. And turns out it's got its own DNA. So there's DNA in the nucleus. Right, so there's DNA in the nucleus, but we just found out in the last couple decades that there's DNA inside the mitochondria, and it turns out that this will even last longer than the DNA in the nucleus. So evolutionary biologists, they really look at this one organelle, and they're trying to figure out, did one bacteria eat another bacteria and whatever. Okay. The important thing that you need to know for the test is right here. ATP is made in the mitochondria. If you want to say powerhouse of the cell, fine with me. Whichever. Peroxisomes. So I already wrote what they do here, detox. So think of hydrogen peroxide, right? You can put hydrogen peroxide on like a cut or something, and it'll detox that, right? Same thing here. So it can make hydrogen peroxide, and that's it. That's what peroxisomes do. So let me tell you something before I move on. Let's go back to the main picture of the cell. This is not a typical cell. This has a little bit of everything. There's two mitochondria, and there's like one peroxisome, one lysosome. But really, in our bodies, there tend to be cells that have lots of one thing or lots of another thing. For example, in your fat cells are not making protein. They're fat, right? So you're not going to really find ribosomes. Why would you need to make proteins, right? Your hair follicles are not making lipids, right? There's not a lot of fat in your hair, right? So, but your hair is protein, right? So you're going to find a ton of ribosomes because that's what hair is. It's protein. So you're going to find a ton of ribosomes, but you're not going to find hardly any lysosomes because that's not the job of a hair follicle, right? You're not eating stuff. But if I were to go to my intestine, then yeah, I'm expecting to find a lot of lysosomes, right? So it's structure fits function.
I got a question.
Yeah.
So when they do a DNA test, it don't show you when they do a DNA test from your hair. it don't show you the difference between the lysosomes and the other stuff.
No, it won't. It won't do it. It's showing you. It's showing you if you're looking at just the DNA. That's all it's showing you. You wondering about drug tests?
Huh?
You wondering like how they do those drug tests with the hair?
Yeah.
I don't know. I wish I knew. Like, why does some stuff like stick in your hair for a long time? You know what? I'm actually going to ask my wife. Hey, Sarah. Yeah. Why do they use hair for like drug tests? What are they looking at? Well, no, they're looking at. She's a pharmacist. She teaches pharmacy. Oh, she doesn't know. There she goes. She's just saying something to shut me up. That's what she does. All right, so we don't know. Yeah, I don't know. Yeah, she's saying she doesn't do it. All right, we're just asking. She's telling me I'm a drug addict. All right, yeah, we don't know. I don't know why they do it. But as far as I know, THC is lipid soluble and not water soluble. And so even with like that type of information, because we already know about like this idea of being mixing with lipids or mixing with water. So already we have an understanding about how it's.
They use the hair because the drugs actually become portion of your hair, especially someone who uses drugs often.
They'll go into the hair?
Yeah, like a urine test only goes back 3 days, but
Yeah.
a hair test goes back more than a little bit of 90. So when they use someone who consistently uses drugs, they'll pull the hair and it actually, like so much drugs, intertwines with your hair.
Oh. So they're probably using like proteins from the drugs and incorporating it into the hair? I mean, I don't know.
Yeah,
I don't
because
think it'll work with everything, right? Like it can't work with every drug. I mean, it works for some drugs,
No.
but not others.
It works with some. It's just some drugs actually lead into your bloodstream. And so when your hair grows out, it becomes part of your hair.
Huh. That's interesting. Definitely nothing to do with the DNA, though.
No, not really.
But I'm not sure that they can detect like weed from that. But I don't know. Who knows? So my question to you, and this is sort of related to drugs, what cells, where would you find cells in your body that have like a whole bunch of these peroxisomes? So I guess my question to you is what part of your body detoxes? I hope you're thinking liver. So in your liver, you would find a bunch of these, right? Because that's one of the functions, one of the many functions of your liver cells to detox. So you would find a lot of these in cells in your liver. I'll get back to that. So we have something called the cytoskeleton. I'm not going to ask you this on a test, but I'm going to just tell you about it now. A cytoskeleton is like, they're like metal bars, like rebar or something like that. They're not metal, first of all, but I'm trying to like give you an analogy. They're like these rods, and we can use these rods for different things. For example, if I look at the cell membrane, right, I can put these rods in there to help give them strength. right? It reinforces stuff and it gives it strength. If you look at the cell here, it's got like this tail. And most of our cells don't have these, of course, but some bacteria, for example, they have like these tails, which we call flagella, and they whip them around like a tadpole, right? Cells have them too. And if you look inside of that tail, you see all these rods. Or even there's something else called cilia. Let me see if I've got a picture of them. Like here. So this is like a bacteria, and you see all these little hairs that it has. We call those cilia. They're actually using those to move around. So they just beat them back and forth and they move around. Things like amoeba, little microscopic things, they can use these cilia and they can move around. Those cilia are made from these rods, right? And so we call this all together the cytoskeleton. So I don't care that you know about this. Next semester in A&P 1, you're going to learn about we're going to talk about microtubules maybe next week, but you're going to learn about like actin and myosin when we talk about how muscle contracts. But for now, I'm not going to ask you about the cytoskeleton. I'm not going to ask you about any of this on any test coming up. So just be aware of cytoskeleton. I will use the word microtubules, but I'm not going to it's not like a test. It's nothing I'm going to test you on. So, yeah, cilia and flagella, that's how cells can move around. The vast majority of cells don't move, but some do. Okay, microtubules. Well, I'm sorry, not microtubules. Centrosomes, right? Centrosomes, I didn't write the word here, but they're used in mitosis. So we're going to talk about mitosis next week. And mitosis is, we're going to talk about something called the cell cycle, how one cell becomes two cells. That's all the cell cycle is. Part of the cell cycle is that you have to take one nucleus and split it into two nuclei before you can split the cell in half. And before you can take one cell and make it into two, you have to take one nucleus and make it into two nucleus in the cell. That's what mitosis is. This is involved in it. So I call them centrioles. Like the words are used interchangeably. They're not technically, but like a lot of people will say centrioles or they'll say centrosomes. And they do the same thing. It's involved in mitosis. So just put those two words together. Centrosome or centriole, mitosis. And that's all you need to know. So if I were to ask you a test question, I would say, what organelle is involved in mitosis? Or I would say, what do centrioles do? That's it. You just put the other word. Okay, that is everything on a animal cell. So of course, by saying animal cell, I mean animals and us as well. And I think I covered everything that I want to cover. Yeah. So we went over all of these. Ribosomes make proteins. Golgi apparatus repackages proteins. Mitochondria, ATP is made there. Lysosomes, digestive enzymes. Peroxisomes, detox. Centrosomes, mitosis. Right, so here is a plant cell. There are some differences here. There's 3 or 4 differences. But the first difference and the most noticeable difference is you see here in white, that's the plasma membrane, but there's also a cell wall around it. So plants have cell walls. We don't. We have a skeleton. Either you have a skeleton on the inside or you have a skeleton on your outside, like insects and and crawfish and stuff, they have an exoskeleton. So either you have a skeleton on the inside, a skeleton on the outside, or if you're a plant, your cells are like little skeletons. They're like little bricks, right? So they use this to help them stand up and to give them strength. So plants have a cell wall. This is, if you remember back to the chapter on carbohydrates, this cell wall is made from cellulose. But anyway, they have a cell wall. They have a plasma membrane like we do, and they have a cell wall. So I was giving like an analogy, like our cell membranes are, they can break, right? They're not super strong. This cell membrane that we have, I gave the analogy of it being a balloon. Like it could get too big and it could pop, or it could shrivel up, right? Plants, they have a balloon inside of a box. So you can blow the balloon up as much as you want, but it's not going to break because it's inside the box. So it'll just take the form of the box. And that's kind of what's happened here. You see this plasma membrane or the cell membrane? It's all the way up against the cell wall. So you can see like that little line right there, right? So it's all the way up against it. So anyway, plants have a cell wall. 2, you notice these green things inside. So those are called, wow, had the name slip my mind. I'm like thylakoid membrane, grana, chloroplast, sorry. Oh, and the word's right here too. Stupid. So cell wall, chloroplast, because plants can plants can make their own ATP. They use sunlight. We make ATP because of food. We eat the food and we take the energy that's in the food and we make ATP with it. Plants, they make their own ATP from sunlight. So this is where it happens. For them, it happens in the chloroplast. So they have a cell wall, they have a chloroplast, and number 3, I'm just going to leave it at 3. The third difference is that you see they have this big space in here, which is called a vacuole. It's right here. They're calling it a central vacuole. Okay, fine. Central vacuole, vacuole. A vacuole just holds stuff. It holds some waste, but mostly it holds water. So if you ever looked at a plant, like when a plant, like let's say like a tomato plant, right? And it doesn't have, you haven't watered it and it's like wilting, or it's like bending over. And then you give it water and you go check back on it a little bit later, half hour, an hour later, and it's standing back up again. And that's why, because as soon as you gave the plant water again, it filled this vacuole up. And when you fill the vacuole up, it pushes all of the organelles. You see how all these organelles are like shoved up against the cell wall? And it makes the cell wall like bulge out. So when you look at like a tomato plant, if you were to look at it under a microscope, it's like a bunch of bricks just all next to each other. They're packed in there like bricks, right? And each brick is like bulging, right? But if this vacuole were to shrink because the plant doesn't have any water, then all of this stuff outside, all these organelles will get pulled in, right? And it won't be bulging anymore. It'll be like smaller. So the plant starts to tip over. So that's what it does. It's just a big holding place for water. So notice what's not in plants, lysosomes, because there's no digestive. The plants don't eat. So therefore there's no digestive enzymes needed. So anyway, I would like you to know 3 things that are in plants that are not in animal cells. And we just listed them. Vacuole, cell wall, and chloroplast. All these words are like, I'm forgetting them. Plasmodesma, don't worry about it. I mean, I'm not going to test you on it, but if you, as you see here, it's like they're holes. They're holes in between cell walls. That's like a way for a plant to move stuff between one cell and another. But that's just FYI. You don't need that for the test. So, chloroplast, don't worry about the parts of the chloroplast. That's a different chapter. Just, it has a chloroplast. That's where photosynthesis happens. Sorry, I probably didn't tell you that. Chloroplast, photosynthesis. Vacuole stores water. And then the third difference is the cell wall. I don't know what to say about the cell wall. Cell wall is cell wall. I have another chapter, a much shorter chapter I'm going to jump into. Let me stop this recording first.